So far we have studied intragenic interactions like codominance, incomplete dominance, the lethal genes and pleiotropy, etc. Now we have to pass on to intergenic or interallelic interaction. This is also called interlocus interaction. Interlocus interaction. What is the meaning for that one? It is the interaction that occurs between the alleles of two different locus. That is the allelic interaction between two alleles. The interaction between the alleles of two different genes. So, we have to go for the different types of intergenic or interallelic interaction where it occurs between two alleles of different locus. Now, what are the types? We have the following types of interactions: intergenic or interallelic interactions. So, we are grouping under what is called epistatic interactions. So, we have 7 types of interactions given in your book. Number 1, dominant epistasis. Number 2, recessive epistasis. Number 3, duplicate genes with the cumulative effect. Number 4, complementary genes. Number 5, supplementary genes. Number 6, inhibitor genes. And number 7, duplicate genes. Now what are the examples? For dominant epistasis, we have the fruit color. Fruit color in summer squares. This is an example. The fruit color in summer squares is taken into consideration for the dominant epistasis. Here the phenotypic ratio, according to the factor hypothesis, there are different phenotypic ratios are possible. Here the phenotypic ratio is modified from that of the Mendelian ratio of 9:3:3:1. So here we have 12:3:1. This is what is called dominant epistasis. We will study in detail just in the next episode. Now we have to go for the next one, recessive epistasis. Here the example is the flower color. The flower color. That is in snapdragon, that is anti-rhino. So what is the ratio obtained here? The modified ratio is 9:3:4. This is a modified ratio for what is called recessive epistasis. The next one we have to go for, that is what is called duplicate genes with the cumulative effect. Here we are taking the shape of the fruit. Here we are taking the shape of fruit of summer squash. Of summer squash. The same one. The fruit. The first one we are taking the fruit color. Here we are taking the fruit shape. Here we are getting the ratio. Just normally 9:6:1. So this is about in the case of actually duplicate genes. Now for complementary genes, we are taking the flower color. The flower color. Flower color in sweet pea. Flower color in sweet pea. The binomial for this one, Lathyrus odoratus. The binomial for sweet pea, Lathyrus odoratus. Here we have the ratio 9:7. So that's about the complementary genes. In the case of supplementary genes, we are taking what is called the color of the grain in maize. So maize grain color. Maize grain color. So we are taking what we have actually the ratio. That is the same ratio what we are getting in the case of that is recessive epistasis 9:3:4. 9:3:4. Now leave the inhibitor genes. Here the example is the color of the leaf in rice. Leaf color. Leaf color in rice. Leaf color in rice. The ratio is normally is that one. We have 13:3. 13:3. Now the last one, duplicate genes. We are taking the example, I am writing the example here. The example is nothing but the fruit shape, or actually see that one, seed capsule shape. Seed capsule shape. That is always called as a fruit shape. In one variety, what is called shepherd's purse, nickname shepherd's purse. This is a name. The binomial is Bursa pastoris. So this is having a trinomial name. So in the case of the last one, duplicate genes, we are taking the seed capsule shape, that is a fruit shape. The plant is commonly known as shepherd's purse, and the scientific name a trinomial Bursa pastoris. So these are the different types of intergenic interactions. We have in a syllabus, not all. We have only one study, one example, that is about dominant epistasis. We have to study the fruit color in some class in details related to the dominant epistasis, where the phenotypic ratio 9:3:3:1 is modified into 12:3:1. Examples for intergenic interactions is dominant epistasis. What is dominant epistasis? Two pairs or two alleles of one gene, or we can say two pairs of alleles of a gene at one locus. So I am taking easy that one, the two alleles, maybe heterozygous or homozygous. The two alleles or the two pairs of alleles, we can say, of one gene at a locus interferes or masks or suppresses the phenotypic expression of a pair of alleles. Suppresses the phenotypic expression of a pair of alleles located at another locus. A pair of alleles of a gene located at another locus. That is, here the gene W suppresses the effect of capital G gene as well as small g gene, because the W is concerned with epistatic gene, whereas G, capital G, the dominant, the small g letter, just lowercase, referring to the recessive allele, and both are being masked or suppressed by what is called the dominant allele. I'll repeat once again. A pair of alleles of a gene, their W, at one locus, say an example of any locus, interfere or mask or suppresses the phenotypic expression of another pair of alleles of a gene, and that is called, actually that gene is located in another locus, is called what is known as dominant epistasis. Now the gene that interferes or masks the phenotypic expression of another allele at another locus is called epistatic gene. Here the W is called as epistatic gene. Whereas the gene whose phenotypic expression is interfered and which prevents the exhibiting of the character is called hypostatic gene. Now both capital G and small g, the dominant and recessive alleles, are called hypostatic gene because whose expression normally suppressed by the dominant gene W. So a non-allelic gene, that is capital W, suppresses the effect of another gene present in another locus. Here W is present in one of the homologous chromosomes at a particular locus. And we see the gene for yellow or green located in another chromosome at a different locus. So these two are non-allelic genes. That is W and G are non-allelic genes. Now the W gene is the epistatic gene, the dominant allele, because it suppresses the expression of either capital G or small g. Both the dominant and recessive alleles responsible either for yellow color or for green color are being suppressed. This is called what is known as epistasis. Now we have to go far. I mentioned already in the case of summer squash, the fruit color locus has a dominant gene, that is W, which is for white color. So the dominant allele for fruit color locus is capital W, or the dominant one, that is concerned with white color, whereas its recessive allele produce colored fruit, any colored fruit, simply we can say colored fruit. So dominant allele for white fruit and the recessive allele W is for, that is, colored fruit. Similarly, the gene capital G, the dominant allele, is concerned with the production of what is called yellow color only in the absence of dominant allele W. Dominant allele W. So that's why G is called as the hypostatic gene. Either the dominant gene G or the recessive gene G, and both are actually hypostatic genes which are not being expressed in the presence of the dominant allele capital W. That's the dominant allele. So anyway, now this is the hypostatic gene present in another locus. It's a recessive allele, that is what we call the lowercase small g. Now the yellow color is normally appeared only in the absence of W, that's a dominant allele. And whereas the dominant allele G can express itself as yellow color and suppresses the expression of green color. So the capital G, the dominant allele, being expressed only in the absence of capital W. And similarly, the green color is expressed only in the absence of both W or, that is, small w or, that is, G, capital G. So the green color will be expressed only in homozygous condition, recessive homozygous conditions, in the absence of both the dominant allele W and the another allele G located in another locus. So this is the one. So W is epistatic gene, and capital G and small g are hypostatic genes. They cannot be expressed, they can be expressed only in the absence of WG, that to the dominant allele. Now, what is happening? So, a plant with white fruit, that is summer squash, was crossed with yellow fruit. Now, this is a genotype of white fruit, that is, two dominant alleles, WW, and then two recessive alleles, that is G. And whereas in the case of yellow fruit, we have two recessive alleles for white and two dominant genes for yellow, that is GG. So GG, the dominant allele for yellow. So it can be expressed, you see that one, only in the absence of dominant allele W. That's why it's yellow fruit. Now a cross is effected between pure breeding white fruit and yellow fruit. As a general rule in diabetic cross, the F1 generation, we have received the gametes. So all the gametes are similar, having one dominant W and one recessive G. There we have one recessive W, one dominant G. These are the two types of gametes. So by random combination, we have received a heterozygous individual. I mentioned already one capital letter or a dominant gene for the white color is present. It suppresses the expression of both, that is what is called yellow color and green color. So the resultant F1 is a heterozygous, but it is white in color. So here you can see the masking effect, suppressing effect of the dominant allele W, which suppresses the expression of, that is both, that is dominant yellow and then recessive green alphabets or the alleles. Now what are the gametes produced? As a general rule, you know that one, haploid gametes are produced by these heterozygous individuals, as in the case of brown, yellow, green. I mentioned already how to remember actually the gametes. So the same way we are getting the gametes. So we have to take the first two letters in the heterozygous condition. So the first two letters are one dominant, one recessive with reference to W. Okay, I am writing each one twice. Each one twice. And taking the next two letters alternately. Capital G, small g, capital G, small g. So the mechanism would be the same. How to remember the formation of gametes in a hybrid, that to dihybrid. The same mechanism either for dihybrid, for any processes, though the phenotypic ratio is different, but the nature of the gametes produced will be the same. That is nothing but independent assortment, segregation. So we have received 4 different types of gametes. So these are all the gametes produced, I mentioned already. So we have to go for the random combination of these gametes to form the F2 generation. So what is the phenotypic ratio obtained in the F2 generation? So remember, the dominant letter, that is W for white, is dominant over either yellow or green, that is capital G gene or small g gene. So it occurs the same in the form of a checkerboard. When you have found heterozygous white colored fruits on self-fertilization, they produce 4 types of gametes in equal proportion, as in the case of dihybrid cross. So the gametes they represent just the male gametes and the female gametes, as in the case of dihybrid cross. It's a single multiplication. Now what is the phenotypic ratio obtained here? So I'm writing, this is one genotype. This is another genotype. This is another genotype. So I am writing just in proper box. So capital W, small w, just capital G, capital G. Here capital W, small w, capital G, small g. So this is box number 1, box number 2, 3, 4, as in the case of dihybrid cross for easy understanding. This is box number 2, box number 3, box number 4. I mentioned already whatever genotype we have in the second box in horizontal line or horizontal one, also we are getting actually in the second box. So we have WW, 3 dominant vectors. Likewise here, 3 dominant vectors. Now here, 2 dominant vectors. The same one also you are getting here in the diagonal line, as we have discussed already, the same one. So now we have a C. At least one dominant gene is enough to produce the white color. So here one is enough, one is enough, one is enough, here also one is enough. In all these cases, one dominant gene, the allele W is present, that is enough to produce a white color. Now let's see what are the other genotypes. Now here I am taking, this is once again, this is one genotype. And here another genotype. And here I am writing what is called actually the same genotype. Now this is also having at least one dominant allele W. So it can also produce what we have, the white flower. So all these 12 individuals, what I have presented, so here and this one all the blue colored ones, so the 9 in the form of triangle and 3 just in the apparent triangle together produce what's called the white. So what is the phenotype? What is the phenotype in F2 generation? 12 white. So what about this one? So we are taking actually, you see the recessive gene W, but 2 dominant genes. And here we are writing just 2 recessive genes. I am taking a different color for understanding. So I am taking this one. Two dominant genes. 2 recessive gene, 1 dominant gene, capital G. And here also we have 1 dominant gene, capital G. See here, in the absence of dominant allele W, now the gene for yellow color is expressed. So this is yellow because at least 1 capital G is responsible for the expression of yellow character. Here there is no white color dominant allele. So the hypostatic gene capital G is expressed in the absence of epistatic gene, what is called the W. When W is present, here you see that one, the expression of the yellow color is suppressed. Here you see that one dominant gene, W is there, so it is epistatic gene. Now capital G is there, but its expression is suppressed. This is because of the epistatic gene dominant allele W. So we are getting the phenotype, the next one, 3, that is yellow, 3 yellow. And what about the last one? As a general rule, in all cases we are receiving at least 1 recessive character. Here, just I am taking and that one, just we see that one, all the genes are recessive, both W and the G genes are recessive in nature, and that one represents what is called green. So we are getting the phenotypic ratio of 12 white, 3 yellow and 1 green. So it is a modified ratio of Mendel of 9:3:3:1. So here the massing effect of that is a dominant allele W, and the hypostatic effect of that is what we have G, but the G gene is dominant over the green. The G gene is dominant over green. That's why the green color is expressed in the absence of either that is white gene or that is what we have the yellow gene. So this is the one what we obtain. Now the dominant allele W is the dominant allele. So the white color dominant parent suppresses the expression of both yellow and green. Now what is the conclusion? Now the homozygous recessive, that is small letter W, produce colored fruit in the ratio of 4 out of 16. What I mentioned color, either green or yellow or both. Now here we see that one, these 4 are colored varieties, that is either yellow or green. So homozygous recessive produce colored fruit means 1 that is green and 3 yellow, together 4. It's a general rule, we have to conclude. So homozygous recessive produce colored fruit, out of 16, 4 are colored, the remaining 12 are white. Then the double recessive, that is the last one, produced green. And this is a general statement. Homozygous recessive produce colored fruit, that is 4. You see, in all cases we have homozygous recessive gene. See, homozygous recessive gene, that is W. That's why I can say there is 4 together. When it is present in double recessive form, it produces green, that's 1 out of 16. Plants with only G. Now these are the plants with only G. Either just we see that one, 2 letters or only 1 letter. Capital G dominant in 2 or 1. Here also 1. At least 1 capital G. In the absence of what is called the white colored gene, produce the yellow color. So plants with only G, not dominant allele W, produces what is called the yellow color. I mentioned, you see that when there are 2 types of genotypes, plants with only G, that is a yellow color. One with what is called capital G here, or 2 dominant letters together in the presence of 2 recessive letters produce yellow. So plants with only G, either actually this form, that is 2 dominant letters, or this form with only 1 dominant letter G, produce what is called yellow color. So these were the observations made in the case of dominant epistasis in summer's class. Okay, so again the phenotypic ratio is modified from 9:3:3:1 into 12 white, 3 yellow and 1 green. Okay, that's about dominant epistasis. So here Y gene is a dominant allele. Epistatic gene suppresses both, that is green and then yellow alleles. So that it is called actually hypostatic gene. G is called as a hypostatic gene and then W is called as epistatic gene. As the dominant gene expresses its character, it is called dominant epistasis.